Hi guys, it's Ashley, and this is my second part of my ILS video. This is my lab number 1, making the plant cell slime. And unfortunately, I had to get a kit where I had to make my slime, and I thought it was going to cover the whole thing, and it didn't. So I'll make do with what I have. So for the slime portion, this is my cytoplasm, which is the jelly-like liquid inside the cell where the organelles excuse me, are found. The cell membrane, that will be on the inside, and I did not paint the inside. I don't know if I would paint it really. Just tell them that the inside is the thin flexible barrier surrounding the cell, and the cell wall is on the outside of your plant cell, and it is the cell membrane. This is the organelle that gives the plant structure. Animal cells do not have cell walls, only plant cells to give it structure. For our nucleus, we will be using a bouncy ball. I did not have a bouncy ball, I have a ping pong ball. So that will be going in my slime for the nucleus. We'll keep it there, hopefully it'll stay. And for the nucleus, this is the organelle that contains the DNA for the cell, which is the genetic material of the cell. And then for our mitochondria, we will be using glitter. And the mitochondria is considered the powerhouse of the cell. This is where sugar is used to produce energy for the cell. So we would just add glitter to our slime for the mitochondria. For our lysosomes, we use buttons. And I will just add a couple buttons for our lysosomes. And the lysosomes are a membrane-bound organelle that is like the trash and recycling center of a cell, and it helps get rid of the waste. So we would describe the lysosomes and add those in our slime. Hopefully nothing falls out onto my floor. And then for our vacuoles, we will be using pony beads, and we will just add some of those to our slime. I'll push those down. And for the vacuole, it is a membrane-bound organelle where fluids and other nutrients are stored for the cell, and it is like a reservoir. So our pony beads are in there. I added a few of those. And then for our chloroplast, we will be using cotton balls, and this organelle is where photosynthesis takes place. And we will add those to our slime. And then if I was in the classroom making this, I would either make students make note cards, or I would make a printout of what each item represents in our slime, and I would tape it to the bottom so they will always have an item to go back to and look at it and say, oh, this is the nucleus, this is our chloroplast, this is our vacuole, mitochondria, and they will just have that visual, and it's just like a fun little hands-on activity for them to do just to learn more about plant cells.